There are about 50 ongoing trials that are actively recruiting right now, 15 of which are mantle cell specific, and only 5 of which are non-BTKI based, only 2 of which actually include patients who've progressed on a prior BTK inhibitor. Oh, that's bad, right? And this is, if we move from the chemo space or the novel agent space into the CAR T space, we've only got one contender in the CAR T space that is targeting mantle cell specifically. That's the Kite Gilead CAR T study. Everybody else has kind of lumped them in. And so are we going to have meaningful endpoints from the other 45 trials, from the other CAR T trials? Or is this going to be one of those, we think it might work, we saw in the 5 patients or so that we enrolled that there was this much activity or this little activity? How do we grow from that? One of the first things that I encountered when I wanted to study or chose to study mantle cell lymphoma, chose to focus on mantle cell lymphoma, was just that. How do I tease out from all these novel agent trials where there's a signal of efficacy when they enrolled 2 patients, 3 patients, 5 patients? And the answer is, I couldn't. There was no meaningful signal. If you saw 2 out of 5 people respond, you had no idea what that meant. 3 out of 5, is that better? I think it's better, but what does that mean? So if we really want to push the bar forward, if we really want to develop novel therapies in mantle cell lymphoma, we're going to have to figure out how to carve out mantle cell lymphoma specific trials, not try to bundle them into, these other groups. Otherwise, we're going to have that same clinical heterogeneity, biological heterogeneity play out with mixed endpoints that aren't going to inform anything in the clinic.